All right, good afternoon, everyone, and welcome to the latest edition of DFHCC's Connect Science seminar series. It's my distinct pleasure to introduce Dr. Sean Morrison, who's going to be telling us how and why stem cell niches aren't just for growth factors anymore. Dr. Morrison, probably well known to many of you, his laboratory studies mechanisms of stem cell function in adult tissues, and specifically the ways that those mechanisms may be hijacked to enable tumorigenesis and metastasis. Sean did his PhD training with Irv Weisman at Stanford and a postdoctoral fellowship with David Anderson at Caltech. Immediately thereafter, he joined the faculty at the University of Michigan, where for about a decade, he directed the Center for Stem Cell Biology. Since then, Sean has been director of the Children's Medical Center Research Institute at UT Southwestern, where he holds endowed chairs in both pediatric genetics and pediatric research. Sean is an investigator of the Howard Hughes Medical Institute since 2000. And he was elected in the last couple of years to both the National Academy of Medicine and the National Academy of Sciences. Sean, it's a distinct pleasure to have you here with us today, and we look forward to hearing from you about stem cell niches and what they're really there for.
Thanks, Ramesh, and thanks for the opportunity to give this talk. Let's see. Before I get started, a few disclosures. I am involved in a handful of biotech companies, including Cogent, Garuda, Ona, Frequency, G1, and Inception Therapeutics. But I'm not going to talk about anything related to these companies or their products today. My laboratory, well, as you all know, you have stem cells that persist throughout life in many different tissues, and that you need to persist throughout life in order to maintain the regenerative capacity of those tissues. We focus on the bone marrow, where there's two kinds of stem cells: hematopoietic stem cells that you may need to make new blood cells every day, and skeletal stem cells that you need to make new bone cells every day. And so we study the intrinsic mechanisms within stem cells that allow them to execute self-renewing divisions, as well as the extrinsic mechanisms by which the niche regulates the maintenance and the activity of these stem cells within the bone marrow. We also compare and contrast stem cell self-renewal with cancer cell replication. And in the context of cancer, we particularly focus on metastasis because it's been known for some time now that cancer is a disease of dysregulated self-renewal, in which cancer cells hijack the mechanisms that normal stem cells use to self-renew and then overactivate those mechanisms to make tumors. So what's of more interest now that point has been made is to identify ways in which the replication of cancer cells differs from normal stem cell self-renewal. And it's for that reason that we focus on metastasis, where we hope that by studying metastasis, we'll learn things about cellular replication that complement the things that we learn from studying normal stem cell self-renewal. So having set the larger stage for what my lab does, today I just want to focus on our work on the niche within the bone marrow. This is a cartoon schematic of the bone marrow, including the arterioles through which blood flows into the bone marrow, sinusoids through which blood flows out of the bone marrow. And this is the bone marrow cavity that you see throughout most of the slide. But this is the interface here between bone and bone marrow, and this is where the osteoblasts live. Almost 20 years ago, my lab discovered the SLAM family markers, or at least that they could be used to simplify and improve the purification of mouse hematopoietic stem cells. And one of the consequences of that is that it became possible for the first time to cut sections through hematopoietic tissues to see where the stem cells localized. When we did that, we found that hematopoietic stem cells were immediately adjacent to sinusoidal blood vessels, which are a specialized form of venous vessel that's only present in hematopoietic tissues. The stem cells are adjacent to the sinusoids, both in the bone marrow as well as in the spleen. And based on that observation, we proposed in 2005 that hematopoietic stem cells reside in perivascular niches created by these sinusoids. Now, the question raised by that proposal was, which cells are producing the factors that are required for hematopoietic stem cell maintenance? And there was really nothing known about that at the time. To make a long story short, we went on and discovered these leptin receptor positive cells that are by far the major source of known growth factors that are required for the maintenance of hematopoietic stem cells, as well as a number of early restricted hematopoietic progenitors within the bone marrow. We showed genetically that hematopoietic stem cells depend on growth factors like stem cell factor and CXCL12 that are mostly produced by the leptin receptor positive cells, but are also produced at a lower level by the endothelial cells that create the wall of the blood vessel. So that proved the existence of a perivascular niche. And in fact, all quiescent and all serially transplantable stem cells depend on these perivascular niches. We went on to show that restricted hematopoietic progenitors reside in cellularly and spatially distinct niches within the bone marrow in different locations. So this was where it got fun, because having identified the parts list, now we could start to do more interesting experiments to try to understand the full spectrum of ways in which the niche regulates hematopoietic stem cell function, hematopoiesis, and osteogenesis. One of the first things that we learned was that these leptin receptor positive cells don't just make growth factors, they also include the skeletal or mesenchymal stem cells in the bone marrow, and that are the main source of adipocytes and osteoblasts in adult bone marrow. Bo Zhou did this work. He's now got his own laboratory at the Chinese Academy of Sciences in Shanghai, where he's from. He showed by fate mapping that the leptin receptor positive cells give rise to adipocyte progenitors that differentiate into the mature adipocytes that arise with age or after myeloablation in the bone marrow. We showed that the adipocytes that arise during adulthood within the bone marrow promote hematopoietic regeneration after myeloablation. I'll come back to that later. The leptin receptor positive cells also give rise to osteogenic progenitors that differentiate into osteoblasts that maintain the skeleton and that repair certain kinds of fractures. Now, there was having identified these leptin receptor positive cells raised an important question in the field, because these weren't the only skeletal stem cells. They're the only skeletal stem cells in adult bone marrow, but there's also a second population of skeletal stem cells that resides on the outside surface of the bones in the periosteum. And the field had kind of divided into camps. There were people like us who tended to think that the cells inside the bone marrow were the important ones, because that's where our data was. And people that had worked on the periosteal cells tended to believe that the periosteal cells were the important ones. And the problem was that there were no markers that were validated to distinguish the bone marrow stem cells from the periosteal skeletal stem cells. And that made it impossible to do a head-to-head comparison of the activities of these cells and which cells were responsible for what. So Elise Jeffrey in my lab took on this problem and did the heroic experiment of scanning, doing side-by-side comparisons of 11 different Cre alleles that various laboratories had published, suggesting that they were markers of skeletal stem cells, to try to identify specific markers of each of these populations. And again, long story short, there were a number of markers that have been the focus of the literature, which don't mark skeletal stem cells in adult bone marrow at all. There were additional markers that mark at least some skeletal stem cells, but they're not specific. They're expressed in both the periosteal and the bone marrow skeletal stem cell populations. And included in this list is the leptin receptor Cre marker that we had been using. We had assumed that this was specific to the bone marrow, and indeed, it does mark all of the bone marrow skeletal stem cells, but it also marks 20% of the periosteal skeletal stem cells. And so the activities that we had seen in the past were at least partly influenced by cells from the periosteum. I should say that leptin receptor is not expressed in an ongoing way in the cells in the periosteum. And so leptin receptor Cre-ER is a specific marker of the stem cells within the bone marrow. But in addition to leptin receptor CRE-ER, we found some additional specific markers. These bone marrow skeletal stem cells are uniformly positive for both leptin receptor and adiponectin. Adiponectin, was first identified as an adipocyte marker, but sometimes stem cells express lower levels of genes that are associated with their differentiated progeny. We see this in multiple tissues, and this is an example of that. We also identified a marker that was specific to the periosteal skeletal stem cells, GLI1 CRE-ER, so that we could use these markers to compare the contributions of the bone marrow cells and the periosteal cells to bone maintenance and repair. So the first experiment that Elise did was just looked under steady state conditions at which cells were responsible for the production in an ongoing way of new osteoblasts. And what she found was that it's the leptin receptor positive, adiponectin positive cells within the bone marrow that seem exclusively responsible for the ongoing production of osteoblasts. And in fact, if you look at where these new cells are arising by fate mapping, they seem to all be coming from the bone marrow side. We weren't able to find new osteoblasts that were being produced from the periosteal side. There's a rare cell that's marked here from the GLI1 CRE-ER mice. GLI1 CRE-ER recombines in 1% of the skeletal stem cells within the bone marrow. So that's a little bit of background, we think. So it's the bone marrow skeletal stem cells that are responsible for steady state osteogenesis. Then we tested a drill injury where we drilled a small hole in the tibia and then allowed it to repair. And what we found was that essentially, or nearly all of the cells that contributed to the repair of these drill hole injuries came from these bone marrow adiponectin positive, leptin receptor positive skeletal stem cells. We saw very little contribution of the GLI1 positive periosteal cells to the repair of drill hole injuries. So if you're keeping score at home, it's the bone marrow cells that are responsible for steady state production of osteoblasts and repair of drill injuries. Then we looked at bicortical fractures. And so if you have a full thickness fracture of a long bone, the first thing that happens is that there's a cartilage callus that forms around the fracture site to stabilize it. And then that cartilage callus differentiates into bone, cortical bone that repairs the fracture site. And then the last thing that happens is that the bone marrow inside the bone at the fracture site, the stroma inside the bone marrow have to regenerate because that gets all destroyed at the fracture site when it first happens. And so we looked at each of these structures to look at the contributions of these two stem cell populations. We found that the bone marrow skeletal stem cells made no contribution to the cartilage callus. Rather, cartilage callus was coming from these periosteal GLI1 positive cells. So now we're starting to see that different fractures are repaired by different kinds of stem cells in the skeleton. We also looked at both, there's trabecular bone that forms inside the bone marrow at the fracture site, also contributing to the stabilization of the fracture, as well as the hard callus that forms to actually repair the cortical bone at the fracture site. And what we found is that the trabecular bone inside the bone marrow came from the bone marrow skeletal stem cells, but the bone marrow skeletal stem cells made no contribution to the cortical bone that repaired the fracture. Rather, all of the cortical bone that contributed to fracture repair came from these periosteal cells. And the periosteal cells didn't give rise to the trabecular bone inside the bone marrow. And so both of these stem cell populations get activated consistently by any kind of a bone injury. And in the context of cortical fractures, both stem cell populations contribute to the initial stabilization of the fracture, with the bone marrow cells making trabecular bone, the periosteal cells making the cartilage callus that then ossifies into cortical bone. And finally, the most surprising thing is that the bone marrow stromal cells that regenerate at the fracture site are not coming from the bone marrow stem, the leptin receptor positive cells inside the bone marrow. they're coming from the Gli1 positive periosteal cells that started out on the outside surface of the bone. So at the late stages of fracture repair, those cells migrate into the bone marrow, and they actually transdifferentiate into new leptin receptor positive cells that repopulate the bone marrow stroma at the fracture site. So normally, hematopoietic growth factors like stem cell factor and CXCL12 are strongly expressed by leptin receptor positive cells inside the bone marrow, but they're not expressed by the periosteal cells on the outside surface of the bone marrow. But when these periosteal cells migrate into the bone marrow after fracture repair, they give rise, they turn off Gli1, the marker they normally express, they turn on leptin receptor, and they begin to turn on these hematopoietic growth factors. So they really look like they're fully transdifferentiating into cells that would normally be creating niches for hematopoietic stem cells and restricted hematopoietic progenitors. So to sum up what I've told you so far, there are two different skeletal stem cell populations associated with adult bones. There are the bone marrow skeletal stem cells that express leptin receptor and adiponectin. They give rise to bone under steady state conditions, and they repair drill hole injuries. But in the context of fracture repair, all they do is to transiently form trabecular bone. In contrast, the periosteal cells are marked by Gli1, at least in the diaphysis of long bones. They're the cells that are responsible for the cartilage callus during fracture repairs, and they give rise to the osteocytes that make an enduring contribution to fracture repair, as well as giving rise to new stromal cells inside the bone marrow. And so the skeleton is fundamentally different than in the hematopoietic system. In the hematopoietic system, we only ever have one stem cell, and whether it's steady state hematopoiesis or regeneration after chemotherapy, that same stem cell population is giving rise to progeny. But in the skeleton, the work is divided over at least two different stem cell populations that make different contributions. So when we had identified the leptin receptor positive cells, we thought that the most important next question to address was whether or not those cells expressed any previously undiscovered growth factors. So Ray Yue, who's now got his own lab at Tongji University, also in Shanghai, did RNA-seq analysis in the leptin receptor positive cells, looked for transcripts that were predicted to encode secreted proteins that were growth factor-like in terms of size and structure, but whose function had never been studied in vivo. And that led to the discovery of a new bone-forming growth factor that we named osteolectin, and that's required for the maintenance of the adult skeleton. Ray showed that it's synthesized by bone lineage cells and by a subset of the leptin receptor positive cells. It maintains the skeleton by promoting the differentiation of leptin receptor positive cells into osteoblasts. Bo Shen subsequently identified the receptor for osteolectin, which is alpha-11 integrin. You probably never heard of alpha-11 because there are very few papers on it. It's highly restricted in its expression to bone-forming cells. And it's required in these leptin receptor positive cells for the osteogenic response to osteolectin. And so if we knock out either osteolectin or alpha-11 integrin in mice, the mice are born in normal numbers. They look normal as young adults, but they develop an early onset osteoporosis phenotype because they just can't make enough bone cells to maintain the adult skeleton. So in the story I'm going to tell you now, we used osteolectin as a marker to resolve some of the heterogeneity among leptin receptor positive cells. To figure out which cells in the bone marrow were making it, Bo, who just started his own lab at the National Institute of Biological Sciences in Beijing, he knocked tomato into the osteolectin locus. And you can see that the only cells here inside the bone marrow that make osteolectin are the periarteriolar leptin receptor positive cells. There are two arterioles in this image that run from top to bottom through the image. You can also see these green structures. Those are sinusoidal blood vessels, and you can see the sinusoids because they're also surrounded by leptin receptor positive cells. But whereas the leptin receptor positive cells around arterioles express osteolectin, the leptin receptor positive cells around sinusoids don't. So this gave us a validated marker for the first time that allowed us to compare the functions of these two subsets of leptin receptor positive cells. And by gene expression profiling, the cells around the arterioles looked like they were poised to undergo osteogenesis, whereas the cells around the sinusoids looked like they were poised to undergo adipogenesis. So Bo followed up on that to actually directly test the function of those periarteriolar cells. He knocked Cre-ER into the osteolectin locus so he could fate map the cells. When he gave tamoxifen to adult mice, it recombined only in these leptin receptor positive cells around arterioles. But the first surprise is that these cells were very short-lived. Within 2 months, the label had disappeared from around the arterioles, and the label traced into new osteoblasts that migrated to the endosteum and that contributed to bone mass. So this showed that these periarteriolar leptin receptor positive, osteolectin positive cells were rapidly dividing short-lived osteogenic progenitors that were a major source of new bone cells. And so one thing that we learned from this is that there was a previously unrecognized niche for these osteogenic progenitors around arterioles within the bone marrow. But the other thing that we had noticed 10 years earlier but never understood was that it's not only the leptin receptor positive cells around sinusoids that express hematopoietic niche factors like stem cell factor. Those factors are also expressed by these osteolectin positive, leptin receptor positive cells around arterioles. And we never understood why the cells around the arterioles were expressing these factors because we weren't aware of a hematopoietic niche around the arterioles. So to explore this, Bo used the osteolectin Cre-ER allele to delete stem cell factor just from these periarteriolar cells. And when he did that, there was no effect on hematopoietic stem cells or multipotent hematopoietic progenitors or most restricted hematopoietic progenitors, but he observed a depletion of common lymphoid progenitors, suggesting that there might be a periarteriolar niche for lymphoid progenitors that was being created by these osteolectin positive cells. So that does seem to be the case because when we localized early lymphoid progenitors within the bone marrow, we found about 1/3 of them were very closely associated with arterioles within the bone marrow. And that's a much larger number than you would expect by chance because there's not so many arterioles within the bone marrow. So deletion of stem cell factor from these osteolectin positive cells broadly depletes lymphoid progenitors, undermining the generation of lymphocytes that are required to clear certain kinds of bacteria. So if we mess up this periarteriolar niche, the mice become more susceptible to listeria, for example. Now, it's been known for decades that lymphoid progenitors get depleted during aging. It's one of the reasons why older people respond less well to COVID vaccines, for example. And others had shown that the depletion of lymphoid progenitors is caused by a change in the environment within the bone marrow. But it wasn't clear exactly what the change in the environment was in the bone marrow because lymphoid progenitor niches weren't well characterized. Bo noticed that the osteolectin positive cells also become depleted during aging, whereas most things in the bone marrow are not depleted during aging. In old mice, we started to see these naked arterioles within the bone marrow that were no longer surrounded by osteolectin positive cells, and it was around these arterioles where we saw the depletion of lymphoid progenitors. So Bo had a brilliant idea at this point, recognizing that the osteolectin positive cells are part of the bone forming lineage, and that load-bearing exercise increases the formation of new bone cells. Bo decided to test whether he could rescue the depletion of osteolectin positive cells and lymphoid progenitors during aging by putting running wheels in the cage with 18-month-old mice. As expected, so mice love to run, and we know that running will increase the thickness of leg bones, and we saw that. We also saw an increase in the frequency of osteolectin positive cells in the bone marrow of the mice that ran. There was no effect on the frequencies of hematopoietic stem cells or most restricted hematopoietic progenitors, but he did partially rescue the depletion of lymphoid progenitors that occurs during aging. And so this suggested that mechanical loading was expanding the size of the periarteriolar niche. And this struck us as important because it's been known for decades that load-bearing exercise promotes the formation of bone cells. It's one of the main recommendations that people get when they get diagnosed with osteoporosis. But the idea had been that the mechanical forces were acting on the bone itself, and that the soft marrow inside the bone was insulated from the mechanical forces. The model for where the new bone cells were coming from was that osteoblasts associated with the bone were experiencing the mechanical forces and proliferating. Now, it was never a very satisfying model because osteoblasts have a limited capacity to proliferate. Instead, these data raised the possibility that the arterioles, which go through the bone surface into the bone marrow, were transmitting mechanical forces from the bone surface deep into the bone marrow, where they were being sensed by the osteolectin positive cells. So to test whether mechanical loading is necessary for the maintenance of these niches, Bo rigged up a way of tethering the mouse's tail to a pulley system on the roof of the cage. So the mice could walk around doing everything they would normally do on their forelimbs, but their hind limbs were unloaded. When we did that, as expected, the unloaded hind limbs became thinner. It doesn't look like a very big effect, but we only let the mice do this for 2 weeks at a time. Within the forelimbs, nothing changed. We saw a much bigger effect in terms of the depletion of osteolectin positive cells within the bone marrow. There was no effect of unloading on the frequency of hematopoietic stem cells or most restricted progenitors, with the exception that lymphoid progenitors were again depleted within the bone marrow. So this showed that mechanical stimulation is necessary to maintain the periarteriolar niche. We wondered what the mechanism was, and Bo recognized that these osteolectin positive cells express the piezo-1 mechanically activated cation channel that Ardem Patapoutian discovered. So to test whether piezo was active in these cells, we sorted out osteolectin positive cells by flow cytometry and into culture. And then Ruma Saeda, who was the postdoc in Ardem's lab who did the first patch clamp recordings on piezo to demonstrate its channel properties. Fortunately for us, she's now a professor at UT Southwestern. And so she did the patch clamp recordings on the osteolectin positive cells. And what she found was that when she applied mechanical force within the patch, she saw an increasing cation current flowing into the osteolectin positive cells, exactly as she had previously demonstrated for piezo. And when we conditionally deleted piezo from the cells, we no longer saw these currents. So piezo was clearly active. We wondered if it was regulating cellular function. And so Bo deleted piezo in vivo from the osteolectin positive cells. When he did that, the bones, the leg bones, got a little thinner. The osteolectin positive cells became depleted within the bone marrow. It turns out that piezo activation allows these cells to divide quickly. And if you take away either mechanical loading or piezo, the cells divide much less frequently and they become depleted. There was no effect of piezo deletion on hematopoietic stem cells or most restricted hematopoietic progenitors. But again, lymphoid progenitors were depleted. So this demonstrated that there was a periarteriolar niche for both osteogenic progenitors and lymphoid progenitors around arterioles, and that this niche is mechanically regulated. Now, we don't know the nature of the mechanical forces that are being transmitted along the arterioles and that activate piezo channel opening in the osteolectin positive cells. But here's one possibility and that I'll animate in this video. This blue vessel is a sinusoidal blood vessel around which there are leptin receptor positive cells that create a niche for hematopoietic stem cells. And this red blood vessel is an arteriole around which there are osteolectin positive cells that create a niche for lymphoid progenitors. Now, these arterioles cross through the bone surface carrying blood into the bone marrow. And so you would imagine that when you engage in movement-induced mechanical loading, that there would be stretching forces that would act on these arterioles either outside of bones or as they cross through the bone going into the bone marrow. And one possibility that's illustrated in an exaggerated way in this video is that those stretching forces that act on the arterioles outside of the bone get transmitted along the arteriole as it goes into the bone marrow, and that the osteolectin positive cells that surround the arteriole, piezo channels get opened as a result of these stretching forces that act on the osteolectin positive cells. And that causes the osteolectin positive cells to proliferate. Since they're the source of stem cell factor for these lymphoid progenitors, the increase in the number of osteolectin positive cells increases the numbers of lymphoid progenitors around the arterioles. And as the osteolectin positive cells increase in number, a subset of them migrate away from the arterioles to the bone surface, where they contribute to the formation of new osteoblasts that accumulate and that thicken the bones in response to mechanical stimulation. So we know that when astronauts go up into space, they lose bone mass and they lose lymphocyte function. And we know that if they engage in load-bearing exercise while in space, you've probably seen images of astronauts on the space station tethered down to a treadmill by big rubber bands that essentially simulate gravity, that engaging in load-bearing exercise at least partially rescues the bone loss and the lymphocyte function. Our data suggests that what's going on when people spend extended periods in space is that because this periarteriolar niche is mechanically regulated, that niche becomes dysfunctional in microgravity, undermining the production of new bone cells from the osteolectin cells, as well as their capacity to maintain and produce new lymphocytes. So I mentioned earlier that adipocytes that get born during adulthood help contribute to the regeneration of hematopoiesis within the bone marrow. And when we first showed that, we showed that the mechanism was at least partly attributable to the production of stem cell factor from the adipocytes. What essentially happens is that chemotherapy or radiation therapy doesn't just kill hematopoietic cells, it also damages the sinusoidal blood vessels that are creating a niche for the stem cells and many restricted progenitors, but it also depletes the leptin receptor positive cells. And the leptin receptor positive cells under those circumstances give rise to large numbers of adipocytes. And the adipocytes become like an emergency niche while the body is rebuilding the sinusoidal niches and regenerating the leptin receptor positive cells. But we felt that we were only seeing the tip of the iceberg and that it wasn't just stem cell factor. Corbin Meacham in my lab, who recently joined a biotech company in Boston, also realized that one of the major factors that adipocytes produce is adiponectin, which is an anti-inflammatory growth factor. And there were kind of competing reports about what adiponectin was doing in the hematopoietic system and whether it was, whether it regulated hematopoietic stem cells or not. So she looked at this question and found that, in fact, adiponectin receptor signaling is necessary to maintain hematopoietic stem cells. And that if we delete, there are two receptors for adiponectin that are expressed in hematopoietic cells, R1 and R2. If we delete both of them, hematopoietic stem cells get depleted. And they will lose their self-renewal potential. They get exhausted during serial transplantation. We wondered what the mechanism was. Again, long story short, Corbin demonstrated that in the absence of adiponectin receptor signaling in hematopoietic cells, there was a dramatic increase in the production of inflammatory cytokines within the bone marrow, and actually systemically within these mice. And with increased levels of interferon gamma, IL-6, TNF alpha. And what's actually happening here is that the adiponectin receptors are required in immune effector cells, including T cells and some myeloid cells. and that in the absence of adiponectin receptor signaling, there's production of inflammatory factors by these cells, and those inflammatory factors in turn act on hematopoietic stem cells and cause an interferon response. Others have shown that when you activate interferon signaling in hematopoietic stem cells, that drives the hematopoietic stem cells into cycle. And so essentially what's happening here is that in the absence of adiponectin and adiponectin receptor signaling, you get chronic activation of the hematopoietic stem cells, and they're depleted. So what this means is, since the receptors are acting in T cells, and that's indirectly leading to inflammation that chronically activates and depletes hematopoietic stem cells, the receptors are acting non-cell autonomously to promote stem cell maintenance. So if we delete either interferon gamma receptor or TNF alpha from these mice, we can partially rescue the depletion. So this is showing the increase in the proliferation of stem cells in the absence of adiponectin receptor signaling. That gets partially rescued if we delete interferon gamma receptor. We can also partially rescue the reconstituting potential of the stem cells. We see the same thing if we delete TNF. And so there's multiple inflammatory cytokines that are contributing to this effect. If we infect the mice with pathogens, that accelerates this effect. So if you give a control mouse sepsis, the mice fights off the sepsis, and the hematopoietic stem cells remain in pretty normal numbers and still have the capacity to give long-term multilineage reconstitution on transplantation into irradiated mice. But in the absence of adiponectin receptors, the hematopoietic stem cells suffer more of a hit to their self-renewal potential, and their capacity to give long-term multilineage reconstitution is compromised afterwards. And so what this is showing us is that adiponectin is partly protecting our hematopoietic stem cell pool from inflammation, and that, you know, we're routinely being exposed to pathogens all the time, some of which we don't even notice, some of which we notice but take for granted that we'll be able to fight it off. But we're absolutely dependent on mechanisms to downregulate the inflammation after these events in order to preserve the integrity and the long-term self-renewal potential of the stem cell pool. And so during aging in mice, normally in a C57 black mouse, the frequency of hematopoietic stem cells in the bone marrow goes up during aging. But in the absence of adiponectin receptors, bone marrow hematopoietic stem cells become depleted during aging, and they have dramatically less capacity to give reconstitution on transplantation into irradiated mice. So I used to think of inflammation as something that happened when something went wrong in tissues, an injury, an infection. In fact, inflammation is like entropy. It just happens. And it happens more as we get older. And I suspect that one of the big things that we'll better appreciate in the coming years in stem cell biology is the need for clonogenic cells to protect themselves from inflammation, not just normal stem cells, also cancer cells. And I suspect there will be many more mechanisms than we appreciate now that are devoted to protecting the stem cells from inflammation. In this case, adipocytes throughout the body, as well as leptin receptor positive cells in the bone marrow, are producing adiponectin. That's suppressing the production of inflammatory factors by T cells and myeloid cells and protecting the hematopoietic stem cells. But that if you delete either adiponectin receptors or adiponectin, you get increased production of these pro-inflammatory factors, which leads to stem cell proliferation and stem cell depletion during aging. So I'll show you some unpublished data. Paul Furnet's laboratory pointed out to the field that there were nerve fibers within the bone marrow. And his lab showed that the nerve fibers are not necessary for hematopoiesis under normal steady state conditions, but that they are necessary to promote the regeneration of hematopoiesis after myeloablation. But not much else is known about these nerve fibers in the bone marrow. It's not known what regulates the innervation of the bone marrow, and there's not much known about the mechanisms by which these nerve fibers promote hematopoietic regeneration after myeloablation. So we decided to look into this question, and peripheral nerves require neurotrophic factors for their maintenance. There's a handful of different neurotrophic factors, but the only one that we could see clearly expressed in the bone marrow is nerve growth factor. And it is overwhelmingly expressed by leptin receptor positive cells within the bone marrow. So this is yet another thing that these cells are doing to regulate the bone marrow environment. These are periarteriolar smooth muscle cells here, and they do express some nerve growth factor, but those cells are so small in number that actually if we delete nerve growth factor from these cells, there's no effect on nerve fibers within the bone marrow. But rather, if we delete from the leptin receptor positive cells, now the nerves get depleted in the bone marrow, and it's partly because we made a reporter. This is just background. These are the cells that express nerve growth factor within the bone marrow. And if we gate on these cells and ask what percentage of them express leptin receptor, it's 90%. So overwhelmingly, the nerve growth factor in the bone marrow is coming from these leptin receptor positive cells. So that in a normal control mouse, you see these green nerve fibers within the bone marrow, they all disappear by 6 months of age if we conditionally delete nerve growth factor from the leptin receptor positive cells. What's actually happening in these mice is the nerve fibers do grow into the bone marrow during development, but they can't be sustained in the adult bone marrow in the absence of nerve growth factor from the leptin receptor positive cells. Now, consistent with what Paul's lab had published, these nerve fibers are not necessary for normal hematopoiesis. Stem cell frequencies of restricted progenitor stem cell activity is all normal in these mice that lack nerve fibers within the bone marrow. But it is necessary to promote regeneration after myeloablation. So we looked into that in a little more detail, and we're really struck that there's this massive increase in nerve growth factor expression 2 weeks after irradiation when the bone marrow is most engaged in regeneration. And then by a month after irradiation, when the bone marrow has largely regenerated, nerve growth factor expression goes back to normal. And if you look at the nerve fibers within the bone marrow, you see this magnificent sprouting of nerve fibers that we think is caused by this increase in nerve growth factor expression within the bone marrow. You just see a lot more nerves in the bone marrow when it's regenerating after irradiation. This sprouting of nerve fibers is partly what promotes hematopoietic regeneration, because in these mice that lack nerve growth factor from the leptin receptor positive cells, you no longer see this nerve sprouting. And the mice just don't have the same capacity to recover after irradiation. There's no effect under steady state conditions. I've already told you, either in bone marrow cellularity or frequency of LSK stem progenitor cells or frequency of HSCs. But a month after irradiation, when normally you should see substantial regeneration of the bone marrow, the mice that don't have nerves in their bone marrow are pretty far behind in terms of overall cellularity, the frequencies of LSK cells, and the frequencies of hematopoietic stem cells. This hasn't been, I don't think, pointed out so far in the literature, but it's not just hematopoietic regeneration that's promoted by these nerve fibers. There's also regeneration of the sinusoidal blood vessels. And this might be kind of a subtle point for people that are not used to looking at blood vessel morphology, but in these control mice, this is what the sinusoids look like after they've largely recovered a month after radiation. But this is what they look like in the absence of nerve fibers in the bone marrow. You can see that they have a jagged appearance. In fact, these blood vessels are not only morphologically abnormal, they're not patent, they leak. And so it creates a dysfunctional environment within the bone marrow. So these nerve fibers are promoting both the regeneration of hematopoiesis as well as the vascular structure. And what's actually happening here is that the nerve fibers are promoting the expression of vascular and hematopoietic regeneration factors by the leptin receptor positive cells, and probably as well by the adipocytes the leptin receptor positive cells make. So in this data that I'm going to show you, the blue bars are the control animals that have nerves in the bone marrow, and the green and red bars are the mice that don't have nerve fibers within their bone marrow. You can see in the control mice, there's a spike of stem cell factor expression and angiopoietin-2 expression and VEGF expression in the bone marrow 2 weeks after irradiation. And as the bone marrow regenerates, in all cases, it comes back down, just like what we had seen in nerve growth factor. And we know functionally that each of these factors, stem cell factor contributes to the regeneration of hematopoietic cells, angiopoietin-2 and VEGF contribute functionally to the regeneration of blood vessels and sinusoids in particular. If you look at the green bars and the pink bars, these are the mice that lack nerve fibers within the bone marrow. And you can see that you no longer get this spike 2 weeks after irradiation in stem cell factor or angiopoietin-2 or VEGF. And so we think that there are multiple factors that are contributing to the regeneration of the bone marrow as a result of nerve fibers within the bone marrow. We can rescue the effect of nerve fibers in the bone marrow by treating with a drug that's an agonist of beta-2 adrenergic receptor. So normally, in the absence of nerve fibers, you get poor regeneration of LSK cells and hematopoietic stem cells. But if we add salbutamol and activate beta-2 adrenergic receptors, now we get regeneration of hematopoietic cells. So we think the nerve fibers are releasing adrenergic neurotransmitters that activate adrenergic receptors. So the question is, well, which cells are really expressing these adrenergic receptors? And it's the leptin receptor positive cells and probably their adipocyte progeny as well that fill up the bone marrow after irradiation. So if we conditionally delete, there's actually 2 adrenergic receptors that are involved in this, beta-2 and beta-3. And if we delete both of them from the leptin receptor positive cells, there's no phenotype under steady state conditions, but a month after irradiation, we don't see the regeneration of LSK cells or hematopoietic stem cells that would normally occur. And in this case, what this diagram is showing is that the blood vessels in the bone marrow are leaky if we delete beta-2 and beta-3 adrenergic receptors from the leptin receptor positive cells. So the leptin receptor positive cells are helping to maintain the nerve fibers, and the nerve fibers in return are secreting neurotransmitters that activate the expression of regeneration factors from leptin receptor positive cells after radiation. So just to finish closing the loop here, if we delete beta-2 and beta-3 adrenergic receptors from these leptin receptor positive cells, we no longer see the spike in the expression of stem cell factor or VEGF or angiopoietin-2 that we normally see in control mice 2 weeks after irradiation. So what I've shown you then is that nerve fibers are not required in the bone marrow under steady state condition, but they promote hematopoietic and vascular regeneration after irradiation. Irradiation increases NGF expression by leptin receptor positive cells and by the adipocytes they give rise to after irradiation, and this promotes nerve sprouting within the bone marrow. And these nerve fibers promote hematopoietic and vascular regeneration by secreting neurotransmitters that activate beta adrenergic receptors on the leptin receptor positive cells and adipocytes, and that causes them to increase their production of stem cell factor, VEGF, and angiopoietin-2. So as a result of discovering these leptin receptor positive cells, we've learned a ton of new biology. We used to think of niches as being the cells that produce the factors that are required for hematopoietic stem cell maintenance. But in fact, these niches have much broader responsibilities for the care and welfare of the hematopoietic stem cells they support. There are different subsets of leptin receptor positive cells that create distinct niches in different locations within the bone marrow for different subsets of stem and progenitor cells. The leptin receptor positive cells don't just make growth factors. They also give rise to the osteoblasts that maintain the adult skeleton, as well as the adipocytes that accumulate during aging. They regulate vascular regeneration. They transduce mechanical forces into niche factor expression around arterioles. They protect hematopoietic stem cells from inflammation, and they sustain bone marrow innervation. So I tried to acknowledge the people as I went through that were involved in doing all these experiments. Many of them have gone off and started their own laboratories in various places. I didn't talk about Jesselyn Ubelacker today, but she just started her lab at Harvard in the School of Public Health. So I'll stop there and be happy to take questions.
Sean, that was a terrific talk with beautiful physiology, molecular biology, genetics, beautiful insights. We're open for questions. Let's see, we have some questions in the Q&A, which, here we go. So let's start with them in the order in which they were posted. So Jonathan Frieden asks, how important is it to take anti-inflammatory drugs, such as NSAIDs, during the acute phase of a bone fracture?
We have
topic, do NSAIDs increase the likelihood of thrombus formation?
Yeah, I don't know. We have not looked at the effect of NSAIDs on any of the biology that I talked about. Ben Levy's laboratory, who is also here in Children's Research Institute, has shown that Ben Levy and others have shown that anti-inflammatory agents do reduce the probability of heterotopic ossification, which is a pathological manifestation of osteogenesis that occurs particularly with highly traumatic injuries. So it does have an impact, but we don't really know what impact it has on the leptin receptor positive cells per se.
It's another sort of casual question whether bone marrow innervation is sensory or autonomic.
It's both. So Paul Furnet's lab, Ioannis Ifantis' laboratory have shown that there are both autonomic nerve fibers, certainly sympathetic, maybe parasympathetic as well, and sensory nerve fibers within the bone marrow.
And then there's a question from Ariel. Is it any increased factors that are involved in feedback between the periosteum and bone marrow in the fracture experiments?
It's a really good question, and that's what we're trying to study now. At this stage, we have this remarkable phenomenon where somehow both kinds of skeletal stem cells in the bone marrow and the periosteum, they both get activated by any kind of bone injury. But somehow they know that one is responsible for repair of certain injuries and the other one is responsible for the repair of other injuries. So there has to be some sort of coordination between them. We don't know what the molecular mechanisms are, but we hope to study that next.
And she also wonders if sonic hedgehog might be playing a role since there's the expression of Gli1.
Yeah, so there is evidence that sonic hedgehog is involved in promoting repair, and we think that's why we see Gli1 expression.
Raul Mostoslavsky wants to let you know there was an impressive talk as always, and he wonders in your osteolectin experiments if you showed that HSCs and progenitor numbers were not affected, but do you know if their function might have been compromised?
Yeah, osteolectin, before we discovered that this was a bone-forming growth factor, there was a total of 15 papers in the scientific literature that mentioned this gene. And people had noticed that it was expressed in the bone marrow. And so it was assumed to be a hematopoietic growth factor. Initially, when we knocked it out, we were hypothesizing that we would see a hematopoietic defect. And so we were highly motivated to find hematopoietic defects in the osteolectin knockout mice and could not find anything. We looked not just at the frequency of hematopoietic stem cells in progenitors, but also at their function in transplant assays and colony forming assays. And so osteolectin is not required for normal hematopoiesis. It's required for maintenance of the adult skeleton.
There's more compliments here. I won't repeat all of them, but people obviously enjoyed your talk. Julio Lay wonders if YAP plays a role in the mechanical stress-induced periarteriolar niche dynamics.
It probably does. You know, there's a lot of people right now that are looking at YAP as being another element of a mechanically regulated pathway that could be contributing to the regulation of bone-forming progenitors. And so we have not looked at this in my laboratory, but there are several labs that have unpublished data suggesting that YAP is involved in the regulation of bone-forming cells in the bone marrow.
And it's perfect time for the last question, whether a adiponectin receptor is required for HSC homing to the bone marrow niche.
We have no evidence that it's required for homing. There could be indirect effects because in the absence of the receptors, it's a more inflammatory environment. And as a result of being a more inflammatory environment, the stem cells are in cycle, but we're not aware of any direct effect. We could not find any cell autonomous direct effect of a adiponectin receptor in hematopoietic stem cells.
And there's a late question that's almost perfect to end a question hour because it allows you to speculate as wildly as you want. And what are your thoughts about CNS involvement in leukemia?
Well, we've not studied that ourselves, but other laboratories have started looking at that. And there is evidence that the sympathetic nervous system in particular plays a role during leukemia progression. Of course, it's not just the central nervous system. These are peripheral nerve fibers, but they are influenced by sympathetic tone that's determined in the central nervous system. So leukemia cells are not that factor dependent. So it's not that you need these nerve fibers for leukemia progression, but there is an interaction between the leukemia cells and the nerve fibers.
All right, on that note, it's great to end on time. You probably have a 1 o'clock Zoom call, as many of us do. So again, thank you so much for spending the lunch hour with us and sharing this beautiful work.
Thanks, Ramesh. Thanks for the opportunity to do it. I appreciate people that made time to participate.
Great, thank you again. Bye-bye.
All right, thank you. Bye.